Hello, my name is Karah Shirley, and I am going to be a senior at the University of Minnesota. And this summer, I participated in the NIH SenNet CUSP summer internship program at Laura Niedernhofer's lab at the University of Minnesota. My main project this summer was doing a single-cell RNA sequencing on fixed dissociated human liver. The objective of this project was to accurately profile transcriptome of fixed human liver samples at a single-cell resolution to identify characteristics of senescent cells. First, I just wanted to give a brief overview of senescence, and I learned a lot about senescence this summer and how there is still a lot to be uncovered. It is currently described as a stable cell cycle arrest that develops in response to stress or damage. The figure on this slide shows that there are 9 hallmarks of senescence, and these genes are really useful when it comes to doing single-cell RNA sequencing experiments to be able to classify different cell types and to understand if they are showing characteristics of senescent cells. For this experiment, we had 8 tissues, and they had an age range of 30 to 60 years of age, and there is a pathology and histology score for each tissue as shown on this table. And most of these samples are normal. However, we also included a couple of samples that are disease as a positive control. On the right, you can see the H&E and trichrome staining of these tissue microarrays, or TMAs. The TMAs contain 4 samples that can fit within the slide constraints of the spatial platforms that we use at the University of Minnesota. The H&E slides show the histology of the tissue sections, and the trichrome staining is used to identify fibrosis, which is stained in blue. Next, moving on to methods, we first had to start with sample preparation, and there's kind of 2 parts to this. But for the tissue fixation, when the tissues were originally collected, they were frozen in OCT, which is something our lab has never done before. So we had to kind of make a new protocol to do this to make sure that we could get fixed cells in the end. And the OCT was removed by washing, and the tissue was fixed overnight in paraformaldehyde. This resulted in a fixed tissue pellet. The next day, the dissociation protocol was started, and the octodissociator was used for this process. Next, the pellet was washed, and after these steps, we had fixed cells, and we could continue with the 10x protocol to complete our single-cell experiment. This is the overall chromium fixed RNA profiling workflow, and this workflow uses fixed samples that are first hybridized with probe barcodes. Next, using a microfluidic chip, the hybridized samples are partitioned into gel beads in emulsion, which are shortened to gems. Inside the gem, probes are ligated and a 10x barcode is added. The barcode and ligated probes are then pre-amplified in bulk, and the gene expression libraries may be generated. This protocol is very useful because, as you guys can see, you can take 4 samples and pool them together since they have barcoded probes, which makes it much more efficient. Also, these probe kits recognize 18,000 genes, and instead of having to make cDNA, the probes attach directly to the RNA, which also makes it a lot more efficient. A little more information on this protocol. The figure on the left shows the probe hybridization and ligation process, where there's a left-hand side and a right-hand side probe for each targeted gene. These probes are homologous to RNA right next to each other, and when they bind to their sequence pair, they will be ligated together since they are in close proximity, and then they also have that 10x barcode attached to it. Then, similar to other methods of 10x RNA-seq, the figure on the right shows how gems are generated by combining barcode gel beads and a master mix of pooled cells and partitioning oil onto the chromium chip. The RNA from the individual cells will be divided into their own droplets and barcoded for that unique cell. So after all that work, we did a lot of sequencing and we got our results. First, we wanted to see how our samples clustered since we did kind of have that difference in age and we had a difference in disease. And as I mentioned earlier, we had one sample that was more disease than the others, and it had a 35% steatosis score. And if you can see these pink areas here, they kind of clustered into their own cell type, which was interesting. And then we also wanted to look at P16 or CDKN2A expression in these cell types. And we also saw some highly expressed P16 in those areas as well, which is interesting. And this is something that we want to look into more in the future and try to see if there's some difference in this disease sample versus the normal. Next, we looked at cell type clustering, and the UMAP on the right shows the cell clustering based on their gene expression profiles. The plots show a variety of cell types found in the human liver, such as hepatocytes, Kupffer cells, endothelial cells, and others. The figure on the left shows the marker genes used to perform cell phenotyping. For example, KLRD1 is a marker gene for NK cells, and this cluster here has very high expression indicating the NK cell population. Also, we can see a very separated cholangiocyte population that is very high in expression for HNF1 beta. Next, on that same UMAP as before, so this is separated by cell type, we wanted to look at P16 expression again because it is such a widely used biomarker of senescence. And the dark red spots shown are likely to represent cells with high P16 expression. And in this one, we can see it kind of here and here and here. And these populations were the monocyte-derived macrophages, the T cells, and the liver sinusoidal endothelium cells. This information tells us that these might be areas where we should look for senescence, and they should be cell typed further in the future to look into that. And then finally, we wanted to compare our single cell data to the SenU of M gene list. And this is a gene list created by the University of Minnesota to study senescence. And it is kind of just common genes that we see and that we study. So we can see this here on the x-axis. And then on the y-axis, we have our cell types that we found in our single cell experiment. And the dot size represents the fraction of cells within each cell type expressing the corresponding gene. And the color intensity reflects the average expression level of the gene in the corresponding cell type. So first, we have this one here, galactosidase beta 1. And it encodes the enzyme beta-galactosidase, and presence of this enzyme is one of the most used biomarkers to identify senescence. We also have the Jun proto-oncogene that is involved in controlling cell proliferation and can influence the expression of genes involved in cell cycle arrest and senescence. Then down here we have MIF, which is a pro-inflammatory cytokine involved in cell proliferation. It can also influence the senescence process by modulating inflammatory signaling pathways and influencing cell cycle regulation. Further down, we have CXCL2 and IGF beta P3, and these are components of the SASP factors and components that regulate cell growth and apoptosis. Finally, we have BCL2, which is an anti-apoptotic protein, and it is one of the hallmarks of senescence, is anti-apoptotic pathways. So all of these genes are very important because they do correlate to the hallmarks of senescence, so it's important to look at them, and they're very indicative of having presence of senescence in our sample. And as you can also see, there are some cell types that have multiple of these genes present, like the cholangiocytes, the DC cells, and then the HSC cells and the hepatocytes as well. Finally, I just wanted to acknowledge my lab and my mentor, Beth Schmidt and Sam Peters, who did all of the bioinformatics and figures for this project, and then the NIH SenNet Consortium, Kay and Elizabeth, for all of their help this summer. This internship was super helpful for me because when I came into it, I had no idea what any spatial platform was or how single cell worked. And I feel really lucky to have been able to learn this because I really do think that it is going to be a big part of senescence research in the future. And it is very novel research, which is really exciting to be able to be a part of.